morning and welcome to everybody joining us today for our Spotlight Series. This is a monthly webinar series by the CTSC of Northern Ohio. My name is Laura Parent. I'm the Director of Strategic Partnerships and Research Development. Today, our topic is speed, strategy, and scientific visibility. For those of you who are joining us for the first time, the CTSC is a collaborative with 7 institutions. Our goal is to increase research, collaboration, community engagement, and train the next generation of researchers. In this session, we are pleased to welcome 2 of our very own from the CTSC of Northern Ohio. We have Roberto Alers-Velasquez. He is our Senior Scientific Editor, as well as Sue Marasco. She's our Assistant Director of Proposal and Research Development. So thank you both to Sue and Roberto. And we are excited to talk about kind of short publications and voucher funding to kind of get everybody up to speed now that we're back into the swing of fall and our fall semester is coming upon us. So thank you both, and I'm going to pass it over to you.
Thank you very much. I appreciate that. Well, hello. Let me get back to my appropriate slide. Okay. The voucher program exists across most of the CTSAs in the United States. It's developed for short-term funding to support collaboration among faculty from different institutions or between faculty and community partners. The role of the voucher funding is to be a rigorous but sort of proof of concept component of a larger grant submission or a larger project. Here are some of the things that you can do with voucher money, which I will tell you more about. But the goal is to get this money, to get it to move things forward quickly, and then to get a product out of it that will support grant funding, which I would love to help you with, or additional parts of the grant or publications. I really loved this graphic. It's from the CTSA out of Virginia. These are 3 authors who are associated with that institution. And this gives you some ideas of what voucher funding has been used for and what it can be used for in each of the translational benefit model products. So it can help develop new procedures or test new procedures. It can look at clinical guidelines. It can be supportive of drug development. It can be part of developing or refining or reusing biomedical technology and software. It can be used in the community health realm to really understand a new way of looking at a community health problem, or accessibility or delivery of services. It can be used in quality of life aspects of how a disease is progressing through a community or through a population. It can work on the commercial entities of developing IP and product development. You can look at cost analysis, cost savings, societal cost of illness for a disease or a process. It can also be used for regulatory policy and scientific communication. So the voucher program we're going into our 3rd year, and in our 3rd year, we really wanted to take the lessons that we've learned from the first 2 years, look at best practices, and really hone in on how to get this process as easy for the PI to submit and easy for the reviewers to say yes to. So we're continuing to anchor some essential parts of it, which are, it does need to be 2 institutions. This is collaborative money after all. So it's going to be 2 institutions or an institution and a community partner is what helps with eligibility. It's a 3-month timeline, so it's very tight. And because of that, you really need to be ready to go when you come to us with your proposal. And it's rigorous. We want this to be, a component of a grant that meets all the necessary components for rigorous research. So we're asking you also to come to the table ready with all of those IRBs and other documentation, regulatory documents ready to go. To help you, once you've got all of those things for us, we really streamlined the process in InfoReady, our system, and we've addressed the most frequent barriers, the pinch points, the bottlenecks that have kept it from being an easy yes on the other end. And we've really worked on making the questions that we ask internally in the application exactly what we want to know to say yes. So, and also the website's been redone so that you just know exactly what you need to do, where you need to put it, and able to do that relatively quickly. And just for us at the CTSC of Northern Ohio, you get to us through the funding opportunities. Voucher program comes up on the voucher program website. We have the voucher application template, which will tell you exactly what the questions are and what you need to answer them. The next step is going to Spark to ask for a voucher consult and all the other lovely services that we have within Spark that can support your projects. Once you've gone to the voucher program and filled that out in Spark, the next step is to put your lovely information in InfoReady. And InfoReady will tell you exactly what you need to do to put the information in. And then it'll come to me and the admin team, and we will be so thrilled to support a successful voucher submission. Once you're in there, just so you know, there are 3 sections to the proposal. We have our eligibility, which is 8 questions that just basically says, are you set to go? It includes 3 document uploads, which are your bio sketches, the IRB letters of approval, and a quote for the cost that you're asking us to fund. The middle section is really the categories of science. Where are you in the translational benefits model? And what does your research include? What kind of study are you doing? What are you proposing? And then the proposal itself is 11 questions, 3 of which are check boxes, and 7 are short answer. And then we also have an image upload where you can put your timeline in all the colors if you wish. So once you've done that and you've gotten your fabulous bit of important money, along with the unwavering and timely support of the voucher team for the next 3 months, we will also hope that you achieve pilot experimental results, community engaged research, proof of concept demonstrations, a method validation, early observational findings, novel data validation, and all of these I want you to come back to me with so we can write a really fundable R01 or an additional mechanism of your choice so that we can get you all the money that you want to do the research that you love. And after you do that, okay, you can do it at the same time. You can also contact Roberto and he can help you turn this lovely little bit of seed funding into a publication. And thank you very much, and take it away, Roberto.
There we go. Okay, let me share my screen. Okay. All right. Can everyone hear and see correctly? Perfect. Let me move.
You're in
this
presenter
away.
mode.
I'm in presenter mode. Is it seeing? Can you see the correct slide now? Like regular
Yes.
slide? Okay, cool. All right. So, as Laura mentioned, my name is Roberto Alers-Velasquez. I am the senior scientific editor for the CTSC. And I just, with this portion, I just kind of wanted to talk to you regarding the benefits of short format publications. Now, this short format publication has been kind of been the discussion throughout the years in the scientific community, as in terms of the debate whether or not, it is useful, whether or not it can help the field. And in 2020, there was a publication where, they essentially did a quick analysis and retrospection on what is short communications and how it is a dying art. So they published this manuscript on the lost art of short communications in academia. And one of the things that they mentioned is that there is a big misconception when it comes to short format publications. And when we think about short format publications, why do they matter? So, well, firstly, it's a rapid peer-reviewed venue where you're able to essentially uh, disseminate your data as quickly as possible and have immediate impact in the field. So these short communications aren't lightweight. They're fast. They're rigorous. They still go through the rigorous peer review as any other research article. And some of these short communications have catalyzed big leaps in the science field. We have the historical DNA paper that it was just 300 words. And that's one of the most famous ones that people know of. And then we have, the 2020 pandemic. So they have propelled big changes in fields with just a short format communication. And one of the things in their analysis that they were discussing, and that they kind of realized, that short communications are an integral part of academic publishing, but because of the misconceptions, their prestige is declining because they don't a lot of in academia, they're not putting that weight under the importance of short format publication. And so there is more to it. There's more to short format publications and the benefits to it. So short format communications can make a huge impact. And I say this because I'm going to take you through this journey on one of the most recent examples of how important short format communications made a great benefit in our public health. So let's talk about the pandemic. So let me show you how short communications changed the tempo of science, and how a handful of concise, rapid updates moved us from the unknown pneumonia into a first in human vaccine in just a matter of weeks. So we start with December 31. The Wuhan cluster was reported. Everything begins here. A brief notice from the WHO and an informal physician alert. So there were no long reports, just enough to essentially mobilize surveillance and bring to the attention of the community of this developing disease. In 2020, the genome was released. In January 10 of 2020, the genome was released. This is a perfect example of how a short format was able to impact the field. So the sequence was posted online, not a full paper. Within hours, groups already started developing and start designing assays and vaccine candidates. So companies like Pfizer and Moderna start sketching mRNA constructs. Then, not too far after, in January 30th of 2020, the New England Journal of Medicine published a letter on the asymptomatic spread. So this is a letter now. This is not a 12-page article, and it reframes the transmission risk. That single page changed the mental model of spread and pushed the early mitigation and trial site planning. So it was short, it was credible, and it was fast dissemination. Then after that, in February, we had the spike structure that was developed and it was published as a preprint in Science. So this is another example of an immediate dissemination of science, where the preprint came first, and then the peer review came not too long after that, and it was able to change the whole dynamic on how they were able to design these vaccines. Then we have the first, because of all these small publications, right? We have the first human dose of mRNA. So this was about 60 days from the sequence to the needle. And that was only possible because of the fast dissemination of data, the regulators, the funders, and all the scientists that were reading these preprints, letters, and short communications and brief reports, all in real time. all the long papers and the detailed mechanisms and all that came after the fact. Then we have the New England Journal of Medicine in March 17, they published a letter on the aerosol and surface viability. So this is another high impact letter, which essentially changed the dynamic on how it's managed at a hospital setting. So it provided a guide on PPE, ventilation, clinical workflows. So it essentially changed the operational aspect of managing this pandemic. Then it was in June of 2020, the recovery, the dexamethasone was released via press briefing. So this is another therapeutic practice, but it's essentially an announcement and that was made and then a preprint. And that changed the practice on how it's employed, which was first done in the UK. And then because of that, it was able to move quick and move over here. So by the mid-2020, the essential vaccine ingredients from all these fast publications, short communications, all of that, were all the ingredients to essentially move the knowledge and speed to combat this outbreak. Now, after all of that, we have that in July of 2020, we already start. So we begin in December, January, and by July, we already have an early phase 1-2 vaccine data. So the pipeline, the brief reports and rapid communications start showing the neutralization of these antibodies. These readouts essentially set up the benchmark across trials to adjust these doses. Then by the fall, November of 2020, press releases on the efficacy of these vaccines start to be released. And by December of 2020, the first authorizations happened. It happened in the UK first, then in the US, and less one a year from the outbreak. And that would have not happened if it wasn't for fast dissemination of data. And by early 2020, we have real world effectiveness and reports on how to combat the variants and how to design the vaccines for those. So this is a good case study on how short communications, preprints, letters, rapid brief reports all did three things. They compressed discovery, they synchronized global teams, and enabled a rolling regulatory decision leading to a vaccine and clinical trials by the end of the year. All this within the span of a year. So what counts as short format publications? Now that we saw how speed can change the outcome and can have real world effects immediately, these are some of the short formats that most journals use when they're publishing. You have short communications. Short communications are usually one clear result, one main figure, minimal methods. These are fast peer review. So usually these are used when finding a solid and actionable, when the finding is solid and actionable and the story is still in progress, but is enough to disseminate and move the field forward. Then you have your research letters, which are very brief, focused, usually a single observation or confirmation that will shift the understanding of the field or practice. Typically for these transmission insights, typically these are used for transmission insights or for early dissemination of safety signals. And then we have brief reports. Now, brief reports are kind of like in between a short communication in terms of how small format they are versus a full research article, which is very descriptive. So your brief reports are a compact study with limited scope. They usually have small cohorts or early clinical trial data, or early phase data. They have a narrow analysis that stands on its own, and they have more context than a letter and faster publishing than an article. Then you have your technical notes. Technical notes are very method heavy. It's usually used when your methodology is the novelty, and that could change the field. So usually scientists publish in a technical note if they want to show their analytical pipeline, or if they want to publish their workflow that can be adopted into different systems or different fields. And then we go into data snapshots, which is usually a just data publication, database publication, where the scientist would feel, or the group would feel, that that is the novelty that will be a stepping stone to move the field forward. One great example from this was when they published the sequence for SARS-CoV-2. And then we have the case reports. Case reports are usually used to present a unique adverse event that will generate a hypothesis or alert clinicians about this particular adverse event. That's usually the common use of case reports, and it's usually a one-page publication. When we look at the short format publications and we put it into a graph, and we think about how complete the data is, and how complex versus how large these documents are, how big the publication will be, you have the shortest, you have your research letters. These are the shortest and least comprehensive. Then you have your short communications, still compact, but with strong results. Then you have your technical notes at the upper left. So again, this is deep, this is very comprehensive data, not a lot of explanation on the actual data, but just kind of publishing the methodology. Then you have your brief report, which kind of really sits in the middle between the complex and the straight to the point. Then you have your case reports at the right lower, which, like I mentioned, is usually used to tell a clinical story. And then you have your full-blown research article, which will have a full mechanism, and it would be complete and would need multiple figures to tell the story. And then one thing that is not mentioned in here, that is not in this graph, is the preprints. The reason for that is because preprints, even though it is a very fast way to disseminate your data, preprints are not peer reviewed. So that's something that is included in the speed layer, as for lack of a better term, and it's very useful. Preprints are extremely useful, and they are accounted for as progress as a scientist, but they're just not peer reviewed. So it's usually you would usually need the feedback of other scientists or the community from that read the preprint, so that they can make a full publication that is peer reviewed. So when we dissect what's the average size of these publications, research letters usually have about 1,000 words. And this depends on the journal. This is kind of like a average of what it would look like for different journals. So the research letter is about 1,000 words. They usually allow you about 2 tables or figures. They will limit you in the amount of references you can use. Like I mentioned, technical note, it would be methodology heavy. So they would only let you about 2,000 words and let essentially the methodology speak for itself. Then you have your short communications, about 2,000 words, 2 to 3 tables or figures, and limit you to 25 references. And then you have your brief reports, which would have a little bit more detail, 2,500 words. They will let you usually to have up to 4 tables or figures, and they would limit you on the references as well, about 20. And then I don't have any descriptors for case reports and data snapshots. They kind of speak for itself. Case reports, like I mentioned, is usually a clinical report. And then a snapshot is a publication of a database for dissemination of any crucial data that can move the field. So when we think as scientists, we think about how could we go through this thought process and know whether or not we can make a short format publication, and where, and if we can. So if we go through this from the top to the bottom, let's say, you have a single clinical relevant claim. If your story boils down to one clear result patients or clinicians care about, it stays within a short lane, and if you need multiple claims to make it work, you're likely in the full article territory. So if your evidence adequate, so you have clear effect of your confidence intervals, and you have all the approvals in place, you would ask yourself, would a critical readers see a signal with reasonable precision and proper IRB approvals. If not, don't force the short paper, just strengthen that first and keep going for a full publication. So if you're not ready, you publish enabling pieces, so a data note, a descriptor, technical method note, or a protocol SAP plan, if the journal allows. So what that would do is that it would timestamp your progression and your contribution in the field and would move the field forward. If your story, your data fits within 3 figures with standard methods, if the main points if the main points you have needs 3 or fewer, then you would go into a short format. If the method itself is the innovation, you would go into the technical method. So because that would be your contribution, and you could write the technical method note. If that is not the case, then you would go into a full article. Then if your data is time sensitive and you need or a priority claim is, so if this is urgent and it's high clinical utility, it has public policy relevance where it could immediately synchronize the field. If that is the case, you would go into a priority or a research brief or a research letter, because that would be rapid editorial handling in most journals, and it would be published immediately or pretty quickly. If it's important but not urgent and it still fits the scope, you target a short communication or a brief report. So usually this how I visualize this tree is kind of a way of thinking about whether or not you have enough to go for a short format publication and have your timestamp of your progression, or keep going and developing your story for a full article. So when we think about in the translational T0, T1 to T2, T4, whether or not you're ready or not for a short format publication, if you're T0, T1, you're ready if you have one claim and you have reported on an effect size with really good confidence intervals and exact n numbers. If you have your baseline and you have really good controls and adequate replicates, the methods are reproducible by 1 or 2 groups, and all the required approvals are stated. If that is the case and your story fits within the limits of figures, then you're ready for a short format publication. If you have no really good controls, no baseline, your confidence intervals are not the greatest. If your methods are a little uncooked or ambiguous, you kind of hold on until that is the more developed, then you can go for a short format publication or a full publication if that's what you would want to target. Then if you're in the T2, T4 space, you have your denominators defined. So how many eligible people or units is set, and you have a baseline. And you have at least 2 points before or after that are feasible. And you can report your reach, your uptake, your fidelity, and include the groups that you're studying. And you have a credible counterfactual control site, and you have developed, you have taken into account interrupted time series or difference analysis. And the outcome is going to be an absolute game changer. Then you should probably think about trying to disseminate that data into a short format. And if you don't have that developed, you don't have your snapshot, you don't have your fidelities lined out, and you don't have proper controls, percentages, counts, then you still need to kind of think about it a little bit more and target that so that you can go for a short format. However, there are some caveats, right? When we think about short communications and we think about this as if you put yourself in the point of view of a scientist, of a faculty, postdoc, grad students, there are some caveats when you talk about short communications. And there's some possible safeguards to that. One of the most important of caveats for short communications is IP protection. So because once you publish the act of publishing your short communication essentially acts as a public disclosure, and it will forfeit any patent rights. So one way that you would be able to kind of safeguard this is to file for any invention disclosure or provisional patent. But you would, you would have to contact your institution's, technology transfer office and IP office to make sure that this is something that you could do. So that way you can disseminate the data and protect your IP. And so this is something to keep in mind as faculties, is that if you are, your research is something that you really want to protect the IP, but you still want to make short format publications, you should definitely look into how you can protect your IP by talking to your technology transfer office. Another thing that can affect your opinion of short communications is for faculty is tenure or promotion. Not every institution is different. Some departments will not see short communications as part of the packet for tenure or promotion. So that way is the one way to safeguard that is to make sure that if you're going to publish a short format communication, that you have your full article not too far away from that short format publication. So make sure that you have a good plan that you would publish the full article, let's say, 3 months, 4 months after your short format publication. And then another worry is scooping. Scooping is it can be very problematic. Is usually frowned upon because if you publish a short communication, you're already putting your timestamp on your program and essentially showing that you are working on this. One way to really combat this, or safeguard this, is to make sure again to align your publication with the full article not too far along, or the preprint with the full submission not too far along. And then we have visibility, the index. The reason why I bring this up is because there's notoriously been cases where a short format publication was the first one to talk about a discovery, and then not too far after, a full research article is published that talks about the same thing, but the research article got more visibility and got more credit than the short communication. And that is usually dealt with by making sure that the journal in which you're publishing has really good index protocols when they're for their articles. And when I talk about indexing, it's essentially how they are able to report that to different databases like PubMed or some of the other like Google Scholars, ResearchGate, all those kind of things. So if if their index is proper, the visibility should be there, and your research should be able to be seen by multiple by the entire field. So, and when I talked about faculties in terms of like the risks, and then for staff scientists, the risk again is making sure that you are when it comes to IP and policy violations, that there is a good transparency when it comes to the particular lab group. So making sure that transparency is there before any IP is addressed or authorship is addressed. For postdoc, this is when it comes to short communications, there's a risk for this, and that is that in the academic field, usually short format publications, when it comes to preparing a package for faculty positions, it's not seen as high impact, and usually they don't put as much weight into short format publications as a full research article. So what, again, to mitigate this is to make sure as a postdoc that you are able to have a full research article not too far along from your short format. And for graduate students, usually the issue is what we call what is referred as the salami slicing. So it's multiple publications multiple short format publications that kind of would dilute the message. So you want to make sure that you align the scope of the research to the advisor, and make sure you have a plan of, like, if you're making a short format publication, that you're able to finish with a punch at the end with a full paper that will really kind of help your career and to get into a postdoc position or industry. So there are multiple different short format outlets that will publish these kinds of articles, these kinds of format papers. You have the Journal of Clinical Translational Science. They publish brief reports. A lot of times here you would go to publish compact single finding with clear translational relevance. You have the Clinical Cancer Research Journal. They publish clinical trial briefs, reports, and research briefs. You have the Cancer Immunology Research and Prevention Research. They also publish these priority briefs and research briefs. We have Frontiers in Physiology, Translational Stroke Research, Clinical and Translational Immunology. They publish their communications, as well as Nature publication. They have a digital medicine where you can publish a brief communication. When it comes to research letters and data snapshots, you have the Journal of Clinical Investigation that they accept research letters. You have the Basic to Translational Science Journal. They publish research letters. You have the Journal of Clinical and Translational Science as well, as mentioned before. They publish letters as well. You have BMC. They publish data notes, research notes, and Nature Scientific Data. They publish data as well. And with that, hopefully that has been of use and useful for all of you. And please, if you have any questions, don't worry. Like, make sure to communicate with us, and Sue and I will definitely help you with all your publication and grant support as we move forward and move the field forward.
Great, thank you both. We are happy to open it up for questions at this time. Are there any specific questions that you might have, or anything relating to the presentation? Go ahead, Shannon.
Thanks, Sue and Roberto. Wonderful talks, both of you. Could you talk a little bit about the changes now that the NIH has changed the AI policy in submitting grants? Could you just speak to that a little bit?
So yes, thanks for bringing that up, Shannon. The NIH has directed the grant submissions to be reviewed for indications of AI. And so it's very important that you keep your research original and that you write your grants so that it's not under particular scrutiny in that regard. Additionally, they're also limiting PIs to 6 submissions a year, including resubmissions. So we also have to be mindful of that new policy. So, but you know, everybody has their secret sauce, and I think the best way to write it is to write that original research into a compelling grant. And we are always here to help you make sure that your data is replicable, it's quality, and between Roberto and I will make sure that what you're writing is compelling and interesting, because what you're doing is compelling and interesting.
And also, just a reminder, I will chime in here as well, is that every Thursday, Roberto offers open office hours. These are really to help answer any question that you might have regarding your upcoming grant submissions, budget justification, manuscript development. So Anything and everything, Roberto, right? What are some
Everything.
examples of questions of support that you've received?
Yes, I've been so things that I've been able to help with is, even if from all the way from abstract development, manuscript editing, to grant editing and review. So I'm able to, if you come in with a question on regarding what what would be my opinion regarding how to put this into a manuscript, or how to put this or just brainstorm on how to make this project into a grant. I'm there to essentially, you can spitball your ideas with me and we can talk about it, and we can come up with a game plan to make this into a grant or publish your data. And one thing that I wanted to mention that I didn't talk about in the presentation is that one of the benefits about short format publication is that, when you're writing for these grants, and Sue can like talk about this as well, is that, when when your grant is getting reviewed as study section, if you have publications in that field talking about, and that could support your project, that will be valued. That will still be seen as, this is a scientist that knows what he's talking about, that know what they're talking about, and is able to be favorable in the scoring. So even if it's a short format publication, like, it's better to put something out and show your progress, and then to kind of draw that. It's always good to draw that line of when is it good enough to stop and let's try to publish something, because as scientists we always want to put more and more and more into this big project and put this marvelous research article out. But sometimes it's good to maybe put an appetizer out and be like, this is what we're working on, this is gonna, expect some more from us. And that will only benefit you as a scientist. So it's something to consider. And please, if you have any questions, please just reach out to us.
Wonderful. And if by chance you need something more intense, so more of a one-on-one, you can always submit a Spark request. So as we wrap up here, just a couple of quick reminders. You can submit a Spark request for either Sue or Roberto under grant development in Spark, in our Spark system, so you can scan there. Of course, stay up to date with our upcoming events, and you can get the link to the open office hours from that QR code on the right side of the screen. So become a member and stay up to date for all of that. And if you would like to explore our next Spotlight series, we are welcoming Dr. Jeffrey Loeb, and he will be discussing UG3 grants and collaboration efforts. So his kind of specialty and background was building AI data sets in neurology. He spoke at one of our partner events at the University of Toledo last fall. And so we are excited to welcome him to discuss kind of this collaboration opportunity a little bit further. So this is a specialty grant kind of in the CTSA realm. So we're excited to have him. And so if you scan that on the left, you can sign up for his session and future sessions of Spotlight series. So we will be, of course, sending the recording out after this. And I saw the request for some slides as well. So we will get those out to you. But thank you all for attending. And now you can go grab your lunch. So thank you all, and we'll hope to see you next time. Thank you.